Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore sickle cell anemia. At the end of this lesson, you will be able to explain the relationship between gene, protein, and phenotype with respect to the HBB gene, or commonly known as the hemoglobin B gene, hemoglobin, and sickle cell anemia. Let's get started. Sickle cell anemia is an example of a single base substitution mutation that causes a significant change in the phenotype. Just a quick revision, we do know that hemoglobin is a globular protein consisting of 4 polypeptide chain, and each chain contains a central iron-containing heme group. There are 2 alpha chain and 2 beta chain make up this a complete globular shape of hemoglobin. So the beta globin gene determines the amino acid sequence of the beta polypeptide. In most individuals, the sequence begins with valine, histidine, leucine, threonine, proline, glutamic acid at the 6th position, and this is coded by the normal allele HBA. However, in the HBS allele, the DNA triplet CTC is replaced by CAC in the gene. This is an example of single base substitution, and this results in the exchange of one amino acid to the another amino acid, which results in the missense mutation. So this changes the sequence of amino acid in the beta polypeptide change to valine, histidine, leucine, threonine, proline, valine at the 6th position instead of glutamic acid. So the diagram here summarizes the effects of the mutation. So due to the single base substitution, the thymine is replaced by adenine, and in terms of the amino acid sequence at the 6th position, glutamic acid is replaced by valine. And due to the substitutions of thymine by the adenine in the DNA, therefore, glutamic acid, which is a negatively charged hydrophilic amino acid on the surface, is replaced by the valine, which is a non-polar hydrophobic amino acid at the 6th positions of the beta chain. So what is the effect in this case? When oxygen partial pressure is low due to the valine, which is a non-polar hydrophobic amino acid located on the surface, there is a reduction or reduce in the solubility, and this defective beta polypeptide chain or the sickle cell hemoglobin tends to form a fibrous aggregate, results in the sickling of the red blood cells. So individuals who are heterozygous HBA, HBS carry the sickle cell trait. Sickle cell trait is not a normal phenotype because in their blood, we still manage to find 50% of their beta globin are normal, and the remaining 50% of their beta globin are defective beta globin. However, they usually do not develop severe symptoms. They may experience mild red blood cell sickling under low oxygen tension, such as at high altitudes or intense exercise. However, individuals who are homozygous for HBS allele, HBS, produce only sickle hemoglobin or produce only defective beta globin, and they develop sickle cell disease or sickle cell anemia, characterized by chronic anemia, recurrent pain episodes, and potential organ damage. Sickle cells have reduced elasticity and can obstruct capillary, resulting in vessel occlusions, which will further reduce the blood flow. And these sickle cells remain deformed even when oxygen level return to normal. The sickling of the cells is due to the fibrous aggregate, where these defective hemoglobin tend to form a fibrous structure rather than the molecular or globular structure. When sickle cells block capillary, tissue downstream receive less oxygen in a scenario known as, or phenomenon known as, tissue hypoxia. Hypoxia and reduced nutrient supply trigger the release of pain mediators and causes local inflammation. So this produces severe pain episodes, and often in the bone, joint, chest, and abdomen. And the pain is sudden, intense, and can last hours to days, sometimes requiring hospital treatment. If vessel occlusion and hypoxia are repeated or prolonged, the affected tissues suffer ischemic damage. So the word ischemic means lack of oxygen. Continuous cycle of hypoxia and reperfusion cause cell death and fibrosis in organ. And over time, this damage can cause chronic organ failure. For example, in kidney, reduced filtration ability occurs, and in liver, hepatic damage from blocked sinusoids may result in the liver failure. Hemolytic anemia is a common symptom of an individual with 2 copies of the HBs allele. The symptom develops because sickle cells are fragile and they can rupture or hemolysis take place after only 10 to 20 days, compared with 90 to 120 days for normal red blood cell. Bone marrow activity increases to replace the cells. However, it cannot keep up with the rapid loss. So in this diagram or light microscope, photomicrograph, we can see that clearly there is hemolysis where the contents of the red blood cells is released due to the rupturing of the red blood cell. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.